Hey everybody, Mr. Grove here. Today we are going to talk a little bit about the DNA story, and so kind of the history behind how the structure of DNA was discovered. And so our first kind of step in this story takes place in 1928. A guy by the name of Frederick Griffith was an army doctor, and he had just went through World War I that finished in 1918, and so he had experienced a lot of trench warfare. Terrible things happened in the trench. That's where people died and got sick and threw up, and just it was a terrible place for disease, and there was a lot of pneumonia. So being a good army soldier, he was trying to develop a pneumonia vaccine that would allow the soldiers to fight and be not affected by pneumonia. And so he did this experiment, and he had one strain of pneumonia. We know pneumonia is a bacteria that was kind of smooth on the surface, so he called this the S strain. Okay, and so the S strain, then, when he injected that into the mice, killed the mice, sadly. Okay, so it was a very harmful strain of bacteria. Another separate strain was rough around the edges of the cell wall, and so he called that the R bacteria. And when the R bacteria got injected into the mice lived, so they were happy. It was not harmful to them. When the S strain was heated, so that's how we kill bacteria, is through heating it. We injected this dead bacteria into the mice, and on trial 3, the mice were happy. And so then his last trial, he took the dead S bacteria, which was fine by themselves, and mixed it with the live R bacteria, which was fine by itself, mixed them up, injected the mixture into the mice, and the mice died. And so that was weird because basically those two things by themselves didn't harm the mice, but somehow that living thing must have changed. And so this is the first case of genetic transformation. So the change in the genotype of one organism by uptaking the genotype of another organism. And we now know that bacteria are very good at doing that. In fact, that's how we utilize bacteria to make a lot of our medicines and antibiotics and penicillin and insulin and things. But then we didn't know. And so the question was, what was Frederick Griffith's transforming material? So fast forward 16 years to 1944, and so that should show you that science just doesn't happen overnight. It takes a while. And so another scientist by the name of Oswald Avery, he was out to try and figure out, okay, what is this genetic material that Griffith used in his transformation? And really, we'd kind of narrowed it down to probably two possibilities, DNA or proteins. And so Avery ran some just kind of qualitative tests, and he noticed the same elements that were in the samples of Griffith were also in DNA. So that kind of makes you think that it might have been DNA. Then he did a chemical analysis, and the ratios of nitrogen and phosphorus in DNA in the samples also worked out. And then he did an enzyme test where certain enzymes would destroy other proteins, and so that should have made the DNA not viable or Griffith's test not work, but they still worked anyway. So that kind of makes you think, well, it must not have been proteins. Maybe it was DNA after all. And so all these things pointed to DNA. Oswald Avery said, hey guys, DNA is the genetic material that Griffith was studying. And amazingly enough, no one believed him. They just thought it was too simple. You know, it's DNA, it's just a G's, A's, T's, and C's. It's too simplistic. Proteins are so much more complex. It's got to be proteins. You know, the basic structure of genetics and genes of life can't be something that is as simple as DNA. So the world moved on without Avery to try and figure out, okay, is it DNA or proteins, even though he was telling us it's DNA. Fast forward 8 more years, now we have Hershey and Chase, and they kind of did their famous blender experiment. So they took the viruses, so we know viruses in bacteria are kind of the little lions and hyenas of the microbial Disney world, so to speak. And so they're always fighting against each other, and sadly, we sometimes as humans get in the way, as we are in COVID right now. But so the viruses would infect the bacteria. And so what we did was we labeled the proteins of the viruses. We know viruses are made of protein coats, and then either DNA or RNA on the inside. Well, this particular virus was DNA. So we radioactively labeled the proteins. It injected into the bacteria. Then we took just a common household blender and blended it up to where the viruses were floating around and the bacteria was floating around then. They were unattached. Then we took a centrifuge, and centrifuge is something that spins very, very quickly. And that centrifuge then, the heavier thing, which bacteria is quite a bit larger than viruses, would slide to the bottom of the test tube and make a little pellet. And so then the viruses would stay floating around in the liquid. And when it was all said and done, where we started with radioactive protein, we still saw the radiation in the liquid, which tells us that the protein stayed with the viruses, so we didn't move the genetic material. A good scientist doesn't just assume, well, DNA must be it then. They actually test it, that DNA would be that. And so this time we used a virus with radioactive DNA. The virus injected into the bacteria. Then we blended it up to get them separated off of each other. Then we centrifuged them to get our bacteria pellet down here, and then our virus floating around in the liquid. And what started off with radioactive DNA ended up with radiation in the pellet where the bacteria was. And this kind of proved it to the science world that DNA is the genetic material. Now, poor Oswald Avery was probably saying, I told you guys 8 years ago, and no one listened. But now the world listened. And so the race was on. We know DNA is the genetic material, and the race was on. What is the structure of DNA? We have to understand structure and how it works before we can apply it to our benefit. And so we had one more minor experiment then. Was done by Erwin Chargaff, and he came up with, in 1949, that no matter where the DNA sample comes from, the amount of G's always equal the amount of C's. And then he also said the amount of A's always equal the amount of T's. And so it doesn't matter if it was a hair sample or from a fish scale or a plant, it doesn't matter. C's have the same percentage as G's, and A's have the same percentage as T's. Now we know with our hindsight that's because an A always matches up with a T. So however many A's you have in that DNA sample, you would have the same amount of T's. However many G's you have in that DNA sample, you would have the same amount of C's. And so that is what we know from our hindsight, but Chargaff didn't really understand why that was. He just knew it was. And so now we're ready for the double helix story. And this is a wonderful story. And these are the people involved. And this was how DNA was found, as far as the molecular structure. And so a lot of my information comes from this book here. This is the double helix story, and it's an autobiography of James Watson himself. So don't take my word for it. Fact check it against the book and see if the story is as true as what it sounds and as cool as what it sounds. So let's start with our guys. So number one, we have James Watson here and Francis Crick. And so this is Watson and Crick. And so Watson, by his own admission, was fascinated with DNA, but was a little bit underpowered. This is not really something he studied, and so he was looking for a colleague then that could help. Okay, and so this wasn't something that he could do on his own, and so he needed to get some assistance, so Crick was the one. So Francis Crick was one of these, by Watson's description. He was kind of one of these guys that kind of knew a lot, was kind of annoying, came up with these big ideas, had you do all the work, and then he wanted to take credit for the ideas a lot. And so he didn't have a lot of volunteers signing up to work with him. But Watson knew he was really sharp and that there was a good chance that he would be the guy. So they joined up to figure out the molecular structure of DNA. The problem is you don't just walk into Cavendish Labs, is where they were over in England, and say, hey, we're here to start working on DNA. There was already somebody doing that, and that was Maurice Wilkins. Okay, so Maurice was already working. He was kind of their boss. And so he would be the supervisor over Watson and Crick, and they worked underneath him. Now, one of the things that we needed to help with our DNA was this idea of X-ray crystallography. Okay, and so we shined an X-ray on a crystal, and then based on the pattern that was produced, it came up with kind of one of these little ink blot looking things. To you and I, that means nothing, but to an X-ray crystallography expert, they can tell, oh, that shape must mean that the molecule was a spiral, which we now know to be true. And so we needed an X-ray, Watson and Crick needed an X-ray crystallography expert, and lucky for them, they had one. And so this was Rosalind Franklin, or Rosie, and that was her area of specialty. Here's where the story starts getting really good. Rosie was the expert. They needed her help. Maurice, their supervisor boss on the program, was madly in love with Rosie. Rosie wanted zero to do with Maurice. All kinds of awkwardness there. I know this has happened in your friend groups before. One friend likes somebody that's also in the friend group, but that friend B doesn't like friend A. And so there's a whole lot of awkwardness whenever your friend group gets together. And so Watson and Crick had to do that dance. How do they get Rosie's help when Maurice is kind of annoying her and bothering her all the time in a romantic, hopeful way? They did that. There's a little bit of controversy in that they kind of took Rosie's information without her knowing it, potentially, or maybe she gave it to him thinking that it wouldn't help. So you'll have to dive into that story a little deeper on your own. But ultimately, they got the information, and they were well on their way to solving the DNA molecule. That's a good story in and of itself, but it gets better. We also have this guy, Linus Pauling. And so Pauling was the American chemist. He had already won a Nobel Prize. He worked for Cal Berkeley. He was the Goliath, and Watson and Crick were the Davids. He had minions that worked for his minions. I mean, he was set. And so he comes out with a giant LeBron James-style press conference. I've discovered the DNA structure, saying that it was a triple helix, and Watson and Crick were like, that is wrong. I mean, it'd be like your math teacher standing up in front of the class saying, the answer to this question, 5 plus 4 is 17. And everybody would be like, what's going on? And so they knew he would be very embarrassed. They knew he would work really hard to try and cover up that embarrassment and come up with the structure. So they knew they had to get going with things to make it happen. Also, if the story ended there, it would still be a pretty good story, but it gets better. Linus Pauling had a son, Peter. Peter was about the same age as Watson and Crick, and they knew each other from kind of science nerd circles. And so as many father-sons kind of go through times where they kind of butt heads a little bit, that was going on with Linus and Peter. And so Watson and Crick was like, man, I wonder what your dad's working on. They're like, and Peter's like, well, you want to know what he's working on? I'll tell you what he's working on. He's doing this. And so they had this little spy going on, and they knew exactly where Linus Pauling was in his research at each level, and so they could kind of know how hard to try on their end. It was almost to the time where Linus was about ready to discover it, and then the story even gets better. He starts having passport visa issues, couldn't get out of the country, and so Watson and Crick swoop in, and in 1953, they give their results to Nature magazine, and they announce that the DNA structure is a double helix with A's pairing up with T's, G's pairing up with C's, and they were right. And so for this amazing accomplishment, James Watson received the Nobel Prize in 1962. Linus Crick, his colleague, received the Nobel Prize in 1962. and Maurice Wilkins, their supervisor, received the prize in 1962. They could not have done it without Rosie, so in 1962 she received nothing. And that is a major source of contention by a lot of people. They think there's a strong possibility that it was because Rosie was female. Now, the Nobel Society stands to their policy. They say our policy is we do not give Nobel Prizes to people who have passed on. And Rosie, sadly, from the time Watson and Crick discovered it to 1962, had died from ovarian cancer. She was dealing with x-rays and probably the safety we didn't have at that point in time. So sadly, she had passed away. So that's their story. The other side says she just got ripped off because she was a woman. No one will probably ever know for sure, so you will have to make your own opinion on that. But that is a great story, and now we know the double helix structure for DNA, and the rest was history, so to speak. At this point, I want you to pause for just 1 second. You can pause the video and get on your phone or computer and Google what year James Watson died. Okay, now you can come back and unpause. And what did you find? Well, in class, students struggle with finding the date he died because as of today, January 10th, 2021, James Watson is still alive. And I tell students, how cool is that you're reading about stuff that happened in a textbook and the person that did this amazing research is still alive. I mean, you could Snapchat him, you could FaceTime him if you really wanted to, and you could ask him the actual story of what happened. We can't go back, and there's no videos of Mendel walking us through his pea garden. There's no interviews on CNN with Thomas Morgan. But James Watson, you could call up and he could tell you the story today. So super cool that we're getting into science that's recent enough that the people who did it are still alive today. So that's the story of the double helix. I hope that was helpful, and I hope it was as entertaining and exciting for you as it is for me. I think it's a great story, and Hollywood should probably make a movie out of it. I think you throw Tom Hanks in there, you could probably have a pretty successful one. So don't forget to smash that like button, and we'll catch you later.